for today? Antibody. I'm going to make you say it till you're blue in your faces. Just kidding. All right. So, after we get done talking again, what are we going to do? We're going to wash. How many times? Twice. Okay. So, again, what would be the problem if we wash too much? We could remove the antibodies, right? And we don't want to do that. And we end up with false negative results. If we don't wash enough, again, we might have that competition thing going on our next step. Because remember, we're creating an antibody sandwich. We're doing antigen, antibody, and then another antibody. And everything's got to be bound. We don't want it free. So then the next step, we're going to add the secondary antibody. So what would our negative controls look like at this point? If we could look down at the molecular level, what should be the only thing stuck in those tubes? What do we have to start with? The chicken protein, which is our antigen, right? So just our antigen, right? Because did the negative samples have the primary antibody we're testing for? Better not, right? That's why they're negative. No antibody. So we're going to add the secondary conjugated antibody. This is your pink tube. If the antibody, the primary antibody is there, the secondary antibody is going to bind to it, right? So for our positive samples, we should have antigen, antibody, that primary one we're testing for, and then the secondary antibody is going to bind to that primary antibody. So what does the secondary antibody bind to? The primary antibody that's attached to the antigen we put in all of our wells, right? That was our first step. What would negative samples look like? Just antigen. No primary, therefore no secondary is going to attach. Pretty simple, right? Not too complicated. After we do the secondary antibody, right, because again, we've got a lot of stuff in here if it's positive, right? We've got antigen, primary antibody, secondary antibody. That's a lot of stuff, right? So we're going to wash how many times? 3 times for that one. Again, we don't want to wash too much. We'll end up with negative results. But we do need to wash enough that unbound stuff gets removed. Doesn't mess up our test. So before the substrate, before I give you the substrate, I'm going to ask you, did you wash 3 times? When we add the substrate, it's an orange tube that I will give you. If the secondary antibody is there, means the primary is there, right? The enzyme will turn over this substrate and we will get the production of blue color. And this will happen immediate. You won't really have to wait around for this. So, let's see. Has it been maybe 5 minutes? Yeah, I think so. So let's, these are some of your questions, right, on your homework. So why is it important not to mix antigen antibody or antigen antibody solutions during the experiment? Or let the solutions from one well overflow into another? What are we doing here in this test? Why are we doing it in the wells? What's everything doing? Sticking and binding, right? We're creating that stack, we're creating that sandwich. So, we need it to react with what on that surface, right? We don't want cross-contamination happening. We need to create that antibody sandwich. Right, so for pregnancy tests, the reason why you get a line, right, is that the antigen or I should say the antibody is immobilized because your hormone acts as the antigen. But you have something stuck to the surface, and then you apply the sample, and it binds, right? Anything that's not bound gets washed away. In the case of the pregnancy test, it just keeps moving off the filter. Only stuff that binds, and again, we've got to have all the components. Antigen, primary antibody in this case that we're looking for. The secondary, which you just added with the enzyme, right? The last step, after we wash away any unbound secondary, is going to be to add that substrate that will give us the color change. So it'll let us know everything is there, right? Antigen, antibodies, secondary antibody. So these are some related techniques that scientists use. You've heard of blot or Western blots. This combines electrophoresis with this. So first you would separate out proteins. Then you would do this step on a membrane. So again, to review, right? Antigen. In this case, it's a chicken antibody, right? And then you added a rabbit anti-chicken antibody. So what they did is they took antibodies from a chicken, put it in a rabbit, and then took those antibodies from the rabbit that it made against the chicken. Then we used another antibody that's going to bind to the rabbit anti-chicken antibody. So they took this and put it into a goat. So now we have goat anti-rabbit antibody. IG stands for immunoglobulin of the class G. Because remember I told you there's 5 different classes. This one has the enzyme HRP attached to it. This stands for horseradish peroxidase. So this compound and hydrogen peroxide is in the sample that I gave you. So guess what? That's why it's in the refrigerator still, that I haven't given you the substrate yet. Because light will affect it, it's also wrapped in tin foil in the refrigerator to stop this reaction from happening on its own. But it's very similar to the catalase enzyme. We're going to generate water. We're going to make changes. We're going to oxidize. We're going to add electrons. Right? Oxidation is a gain. We're going to lose electrons from it. We're going to reduce the other, and we produce that blue color. Right? And we could read this on the spectrophotometer, right? Using a filter of 650 nanometers, and we could actually get a number, right? Just like we got a number at how turbid our things were for Kirby Bauer, I could get a number for how bright. But again, today we're not going to do that. There it is, horseradish peroxidase. Breaks down hydrogen peroxide. So no human stuff, right? We don't have to worry about problems with that. And that's that code. I'm going to leave this up because this is a good slide.